Welcome everyone to this week's edition of the Extracellular Vesicle Club. We have an event today that has been organized in a truly global fashion. So we have people from all over the globe who are joining to present some recent work that was published in the Journal of Extracellular Vesicles, using a technology that allows us to understand a lot about the morphology of extracellular vesicles. And one of the things that has been seen is that some EVs seem to have these multiple layers to them. I was actually just talking with Ute Erdbrugger earlier tonight at University of Virginia, and I think she's on the call right now, and she refers to them as Russian dolls. So, you know, these nested dolls. So we're going to be hearing a little bit about them in this EV Club session, and I'd like to turn over the moderation now to Natalie Turner, who is joining us from Australia. So Natalie, thank you so much for moderating this session, and I'd like to ask you to introduce our guests.
Perfect. So thank you so much for having me to moderate the event today. I'm really excited about this. So we are here to discuss the letter to the editor that was published in February in the Journal of Extracellular Vesicles about these multi-layered EVs. And presenting today will be two of the authors from the paper. So we have Professor Ishi Talmon, who leads the research group at Technion Israel Institute of Technology in Haifa, Israel. He's a pioneer in EV imaging by Cryo-TEM. And then we have our first author on the paper, Kelly Broad, who's a PhD student at the University of Florida Scripps Biomedical Research. And we also have Associate Professor Joy Wolfram from the University of Queensland, who is senior author on the paper. It goes without saying, Ken, you were also on the paper. So we really are joined by, I think, all the authors today. But Ishi and Kelly will be presenting. So Kelly, you're going to start off the presentations by summarizing the letter, and then we'll hand it over to Professor Talmon to discuss the Cryo-TEM and Cryo-SEM from a technical and brief historical perspective. So Kelly, if you're ready, I'll invite you to share your screen and we can get started.
All right, thank you, Natalie, for the introduction. All right, as Natalie said, my name is Kelly Broad. I'm a PhD student at Scripps Research Institute. And previously I worked in the Wolfram Laboratory during a summer research fellowship. Before we get too far into things, I want to say some quick but sincere acknowledgements up front to my co-authors on the letter to the editor. They were truly instrumental to the publication. All the authors contributed to reading multiple drafts of what was my first author work and provided phenomenal feedback and really developed the manuscript into what it is. So I deeply appreciate everyone's role. And aside from his role as an author, I'd like to thank Dr. Whitwer for the opportunity to present an EV Club talk and Natalie for moderating today. All right, to get started, this presentation aims to emphasize how under-researched multi-layered extracellular vesicles are, but first it's important to underscore that identification of morphological EV subtypes would be extremely difficult without cryogenic transmission electron microscopy, or Cryo-EM. This has allowed us to capture images like this. So as you can see, we have the Cryo-EM grid here that is darker due to its density. And then to the right, we have what we would normally think of when we think of an EV, this nice little single lipid bilayer. But next to it is a multi-layered EV that features multiple continuous lipid bilayers. And despite the presence of these multi-layered EVs in vitro cell culture and from biofluids, they remain under-discussed, with only a small number of papers highlighting their presence in samples. Today, my portion of this talk will be split into 3 parts, exploring how isolation and storage methods impact these multi-layered EVs, some identified pathological drivers that change the prevalence of these subtypes, and potential biogenesis pathways. I'll start with summarizing some literature from the last decade. First, we will discuss isolation methods, starting with one of the most common forms of EV isolation, ultracentrifugation. In this publication from 2009, looking at human semen, the authors described a subset of vesicles as high electron density vesicles with an asymmetric deposition of dark substance surrounding one or more secondary vesicles. The fraction of these vesicles in their sample was around 50%, So a pretty high percentage of their sample were multi-layered extracellular vesicles. This work from 2013 examines EVs found in fresh plasma, and very fresh. This was imaged within 2 hours of anticoagulated blood collection. And while they noted the presence of multi-layered EVs, the frequency with which they appear in the sample was not discussed, but it did establish the presence of multi-layered EVs in human plasma. The figure demonstrates morphology we will encounter again, a multi-layered EV with concentric lipid bilayers. Of note, for the second half of this section, this sample was not frozen prior to cryo-EM imaging. This work from 2017 found diverse morphologies of EVs in cultured immortalized human mast cells. They found multi-layered morphologies to be present in relatively low quantities, but familiar morphologies from the prior studies are present, such as the high electron density EV with a smaller asymmetrical EV inside and concentric lipid bilayers. This 2020 publication examined pooled Parkinson patients' cerebrospinal fluid versus controls and found no morphological differences in EVs from either source, but multi-layered EVs were present in both samples. This establishes cerebrospinal fluid as another biofluid containing multi-layered EVs. This publication, also from 2020, is particularly interesting, whereas all the prior publications discussed used only ultracentrifugation. This work is notable for including dead end filtration as well as an isolation method to compare the two. This publication found hippocampal neuronal cell-derived EVs have morphological variability with both ultracentrifugation here and filtration, dead end filtration, yielding samples containing a multi-layered EV population. However, this publication in 2013 found that using dead-end filtration and dialysis greatly reduced the fraction of EVs with a multi-layered morphology. They concluded that cryo-EM sample preparation was not the source of the multi-layered EVs, suggesting that the multi-layered EVs were an artifact from ultracentrifugation, forcing smaller vesicles into larger vesicles. And indeed, when we look at their findings, with ultracentrifugation, almost all the EVs had a multi-layered morphology, whereas filtration, only 7 in 100 are multi-layered, and dialysis, only 2 in 100 are multi-layered. Our letter to the editor included example images of THP1 cell EVs that included multi-layered EVs. However, these EVs were isolated with tangential flow filtration, or TFF. TFF differs from conventional dead-end filtration as fluid flows tangentially across the surface, which avoids the formation of a filter cake. TFF has been shown to be less damaging to EVs compared to ultracentrifugation by previous work done by the Wolfram Lab group. And to note, for the next part of this section, these samples were stored in a cryoprotectant. As we can see, when we look a little closer at these images, the TFF isolated samples contain multi-layered EVs, including some now familiar morphologies. We have concentric lipid bilayers, and then smaller whole vesicles within larger vesicles. The existence of multi-layered EVs as more than an artifact from isolation is not just supported by their isolation via TFF, but also by this publication, which examined unprocessed human ejaculates and found diverse EV morphology. Of particular interest is their total lack of isolation or purification processing. The only process the sample underwent was freezing prior to imaging. The authors of the work identified 11 different morphological subtypes, with multi-layered variants comprising several of these. The authors note that multi-layered EVs represent a smaller fraction of the total morphologies described compared to prior research on purified human ejaculate, which was the first paper we discussed. For a sense of numbers, they identified 853 single vesicles, 165 double vesicles. They categorized those electron-dense vesicles with a smaller vesicle within them as double special vesicles and found 53 of them in the sample, and 39 EVs that were triple or more layers. To move on to our second part of this presentation, unless otherwise noted, the freezing of all of these samples either was not specified what they were frozen in, or they were frozen and thawed in PBS, which is a non-cryoprotectant. And the freeze-thaw cycle in a non-cryoprotectant can cause osmotic pressure changes, the formation of ice crystals, which can puncture and weaken membranes, the detachment of biomolecules of interest, and conformational changes to the membrane and membrane proteins. As such, we wanted to investigate whether storage without cryoprotectants was potentially creating these artifacts. Non-permeating cryoprotective agents such as sucrose that replace hydration spheres through hydrogen bonding interactions help better preserve EVs, as shown in this publication from 2022 on the topic. The concentration and diameter of collected EVs stored in a cryoprotective sucrose buffer remains closer to that of fresh EVs when compared to those stored in a non-cryoprotective buffer, such as PBS. The Wolfram Lab has used sucrose buffer for storage for multiple papers that included images of multi-layered EVs. We see them here, here, here. They're all over the place once we're made aware that they exist and that they're present in our samples. And the publication on cryoprotectants even features multi-layered EVs in both the PBS and the sucrose conditions. To summarize this section of the presentation, multilayered EVs are present in conditioned cell culture media and biofluid. They're present with multiple isolation methods, including ultracentrifugation, dead-end filtration, and tangential flow filtration. They're present in crude samples that underwent no purification and no isolation. And they're present with multiple storage conditions, both in phosphate buffered saline and in cryoprotectants. Now we will move on to discuss potential pathological drivers for multi-layered EVs. This 2022 paper from the Wolfram Lab Group is what initially sparked the discussion of multi-layered EVs between Dr. Wolfram and I. To generate EVs from perturbed monocytes, they were treated with a bacterial endotoxin lipopolysaccharide, or LPS. EVs from the LPS-stimulated THP1 monocytes displayed substantial morphological differences compared to those from naive, untreated THP1 cells. Specifically, multi-layered vesicles with concentric bilayers, such as this one seen in A, were observed at higher frequencies. This is an interesting finding that I'd like people to keep in the back of their mind as we discuss later whether form and function are related when it comes to these structures. There's also noted increases in Gaucher disease, which is a lysosomal storage disease characterized by a buildup of lipids in various cells and tissues in the body. This study did not find a difference in the concentration of EVs in the plasma of Gaucher disease versus controls, but they did note a larger size and more varied morphology, including multi-layered vesicles, in the Gaucher disease patients. The authors of the paper suggest that the release of EVs may serve as an alternative to the dysfunctional lysosome disposal pathway in Gaucher disease. And finally, this publication from Professor Andy Hill's lab group in 2012 noted interesting changes in mouse hypothalamic neuronal cell-derived EVs in prion infection. When prion infected, they noted a reduction of triple membrane vesicles and an increase in single membrane vesicles. They also noted that the single membrane vesicles were similar in size to the interior vesicles in double membrane vesicles. The publication suggests that these small vesicles may be the initial components in the creation of double membrane vesicles and that prion infection interfered with the biogenesis process of these multi-layered EVs. To summarize this section, multi-layered EVs may be associated with several pathological drivers. We note increases in cell culture when treated with a bacterial endotoxin. Plasma from patients with a metabolic disease had noted increases in multi-layered EVs, while there is a decrease in cell culture treated with prions. We will now briefly discuss speculative pathways of biogenesis of multi-layered EVs. To start off, the first three focus on ectosomes, with the first being relatively plausible. There's evidence of intracellular vesicles encapsulating entire membrane-bound organelles, such as mitochondria. So this may be one source for multi-layered EVs. The second mechanism is more speculative, endocytosis followed by membrane budding. This doesn't have to occur right next to each other. It could actually allow for transcellular transport with the EV moving all the way across the cell body. It can also be speculated that the presence of intracellular vesicles in close vicinity to the cell membrane could result in double layer EVs upon membrane budding. And finally, in the case of exosomes, we speculate that encapsulation of smaller intraluminal vesicles and larger ones may be another source, but evidence of this process has not yet been reported. A fifth potential mechanism the letter to the editor was proposed relatively recently for lamellar bodies, which are secretory multilayered organelles found in certain epithelial cells. The proposed model involves flipping phospholipids from the outer membrane leaflet to the inner one through the actions of a flipase, creating perpendicular T-junctions, where the membrane sheets detach, grow, and eventually form curved arrangements of various kinds. Now we have to ask a question of whether form follows function. Multilayered structures are beneficial to synthetic nanoparticles as they allow for the slow, protected, and triggered release of the contents. Multilayered structures also allow for the increased packaging ability of membrane-embedded contents, and the breakdown of these nanoparticles can be stimulus-sensitive or degradation-mediated. If form follows function, it is possible that multilayered EVs may have similar mechanisms for controlled and sustained cargo release and increased packaging of membrane-bound particles. To call back to the perturbed LPS-stimulated monocytes producing increased highly layered EVs, we can speculate that these activated immune cells may be intentionally producing multilayered EVs in order to not only increase pro-inflammatory cargo carried within them, but mediate its release and increase the odds of it reaching its intended target. This speculation raises a question. Does the content differ between and within layers of these particles? The answer as of right now is unknown. The biogenesis of multilayered EVs as a whole remains completely unknown, and it is an area that requires further investigation. To conclude this portion of the presentation, further research on multilayered EVs is needed on their biogenesis mechanisms, on their biological function, and on their relevancy in disease and health. They have the potential in the future to serve as biomarkers for disease, to provide new treatment strategies and new therapeutic targets in the near future. Thank you for your time. I'd like to pass on the presentation so we can discuss further how Cryo-EM allows for us to observe these various morphologies.
All right, very good. Let me share the screen here. Okay, so I would like to, following this excellent presentation, I would like to share with you a little bit, share with you some insight into electron microscopy, cryogenic temperature electron microscopy. This first slide just shows some views of what we see from, not from my home now, but in Haifa in general. So these are the pictures that we have just seen. And the question is, how do we get these pictures, and what are the difficulties, complications, and pitfalls of this technology? So basically, in order to get very high resolution, we use the transmission electron microscope. This is what the transmission electron microscope looks nowadays. It's a big box. Inside we do have, let me show you, an electron microscope. And the transmission electron microscope, and I'm trying in this short presentation not to go into too many technical details, the transmission electron microscope is very similar to the light microscope that most of us know and have used. However, we don't use photons, we use electrons, and because of their very short wavelength, we are able to get down to resolutions that are on the order of 1 angstrom under, or even less, under ideal conditions. The problem is that electron microscopes work under very high vacuum, and in order to make the specimen compatible with the microscope, and there are other limitations of what we can see and what is the specimen in the microscope, we have to make the specimen compatible with the microscope. Before that, here are some classical sketches of liposomes and micelles and bilayers. And the sizes that are involved here are on the order of a few nanometers. So of course we cannot see this by the light microscope. We are able to see this if we make the specimen, the system, compatible with the electron microscope. In the older days, people used what we call staining and drying, which means one used to take people still do it. One used to take the dispersion of your liposomes or EVs, add a stain, typically a salt or acid of a very heavy atom, and then dry it. And in this case, where we look at liposomes that originate from the lung surfactant, we see that the preservation of the structure is really quite bad. We do see that there are some balloon-like structures, but we can say very little about the membrane, for instance. Things are very different when you take this specimen, you make it into a very thin film. I'll show you how we do it, and then you cool it very, very fast. If you do that, you see something like this. So instead of this, you get this. What we see here are very nicely preserved liposomes, and as you can see, even in the lung surfactant, and this is not really a natural lung surfactant, but this is artificial lung surfactant that is used in treatment of premature babies. But you see here too, we can tell very, very clearly. We can tell very clearly that we have single layers and multi-layered and concentric, and your China and your Russian dolls, everything is seen here quite easily. S here designates a support film, and basically what we see here are the vitrified water. I'll talk about this in a minute, and our particles in them. So what we have here is water that is not crystalline, but is really glass-like. And this is really very important, as I will say in a minute. So having convinced you that we have to use Cryo-EM and not staining and drying, let's see how this is done. Before that, let me just say in general, what are the goals of electron microscopy of any soft matter? This is a general technique that we use not only for EVs, but for many others, other small aggregates in liquid, be it water or even non-aqueous systems. So the emphasis is that we get here direct images and we get sufficient resolution, which means we want to get resolution that will tell us what, at the level that we want to know, what is the system like. In some cases, we try to even see how the structure develops. So it's not just a one-point process, but we want to follow a process. And Kelly alluded to it in one of her last slides. And in many cases, we don't rely, as you know, on imaging alone, but we compare it. We compare our imaging data to other data, such as small angle X-ray scattering or NMR. And basically, if we do it right, we get full nanostructural characterization of the system. But it's not very easy. First of all, we have to make sure that we preserve the structure during preparation. The electron beam that we use is a very strong ionizing beam, and if we are not careful, and if we use too many electrons during the observation, we actually destroy the specimen before we can record the image. So this is something that we have to take into consideration. Also, in many cases, the contrast between various parts of the system is insufficient, which means that we cannot tell where the system is if we don't take the right steps to do that. And of course, basically, eventually, we have to take the images with the right camera in order to get an image which has the necessary resolution. This is an old slide which shows very well what happens when you cool your specimen. You get, if it's water, you get ice. This is of course obvious, but however, it depends how quickly you cool your water. If you cool your water really very quickly, you get this featureless glass-like structure, and this is what we want to get when we prepare a specimen. This is called vitreous ice. If the cooling is not fast enough, and this was just a fortunate accident that you see here, we get crystalline ice. This is hexagonal ice, and this is cubic ice. And of course, if the specimen is made into hexagonal ice, then you get all kinds of structural and electron-optical artifacts. So you have to cool so fast that you get vitreous ice. So another problem that we have in preparing our specimen is that we want to make sure that the specimen is prepared at conditions that preserve it before it forms the vitreous ice. So we have to prepare it in some kind of a control environment. And this is a homemade device that we still use. And in this box here, we control the humidity in such a way that we don't lose any volatiles from the specimen during preparation. Also, we can control the temperature in this box. So if we, for instance, want to make sure that the specimen is at body temperature, we can control the temperature in this box to be 37 degrees centigrade. So basically, the procedure is very, very simple. We have a grid with electron microscope grid with a perforated carbon film that you already saw in the lung surfactant specimen. It is held here. This is the schematic. This is the picture here. This is the grid, by the way. You apply a very small drop of your liquid, about 4 microliters. Then you blot most of it in order to form a very thin film. I'll show you in a minute. And then you drop it into freezing liquid ethane, which is cooled by liquid nitrogen. The temperature is minus 183 degrees centigrade. The reason we use ethane is that the boiling of ethane is about 100 degrees higher. So when we plunged our specimen into freezing ethane, we don't get any vapor film around the specimen, which assures us that we get very high rate of cooling rate, which should be for water about 100,000 degrees per second. So this is just a perforated carbon film, the dry one. In principle, what we want to have is that each one of this hole we want to support. Each one of these holes we want to support a thin liquid film. Here is the schematic of one of these holes, and you see this is a schematic of the liquid film that spans on the rim of this hole. And here you can see some of the typical thicknesses that we have in the thinnest areas, 2 to 10 nanometers. In the thickest area, not more than 300 nanometers. This, of course, is a limitation of how big an aggregate we can fit in this film. After we have vitrified it by plunging the specimen into the liquid specimen into boiling ethane, freezing ethane, we load it into a special holder, and we transfer it into the microscope. Now, one of the most important developments in recent years is what we call the phase plate, the Volta phase plate, which is similar to the phase plate that one sometimes uses in the light microscope. And what this does, again, with minimum technical explanations, it transforms phase differences of different areas in the specimen to amplitude differences. This is a picture that is taken without faceplate of a specimen, which is a dispersion of a protein trimer. And what you see here is very little. What you see here is a little speck of ice, and then in the background, all you can see is the vitreous ice. When you turn on the faceplate, the voltage on the faceplate, then you see this. So going from this to that. Again, this is just as a reference. This you can see here, the little ice speck. So basically, we make things that are invisible, we make them visible. And in specimens such as EVs, this is really extremely important. And all the data that we have taken and which Kelly has shown has been taken with these faceplates, which are a very nice device, which you can have on modern transmission electron microscopes. To sum up this part of the talk, let me just show you very, very recent data. And these are EVs that were taken not from our regular cells, but rather from a gram-positive bacterium, Bifidobacterium longum. And this is a part of a very recent collaboration that we have had with another professor, Technion professor, this time from the Faculty of Medicine, Naama Geva-Zadorsky. So we see here the very same kind of vesicles. So what is even more important or more interesting is this thing here. What we tried to do is to try to look at the bacteria themselves as they develop the extracellular vesicles. And possibly this one is the most important of this collage. And what you can see here is this is the bacterium. You see here the two layers of the shell of the bacterium, and here, right here, is the developing extracellular vesicle. And later on, we can also see this, in this case, we see here an area where the EV developed and an EV that is already free. So in this case, one can actually follow or catch, not follow, but catch different steps in the formation of the EV. And this is something that we did before in human cells, as I will show you by scanning electron, when I discuss now scanning electron microscopy. So far we talked about the transmission electron microscope, as I said, the transmission electron microscope is an optical instrument completely analogous to the light microscope, but sometimes we need to use another type of microscope, and namely the scanning electron microscope. The scanning electron microscope, we look in most cases on the surface of the specimen. We scan a very fine beam across the specimen, and at each point scanned by the beam, we collect the signal that is released. And by putting together, collecting many of these picture elements or pixels, we form a complete picture. This is very similar in forming a picture on your cell phone, which is also done pixel by pixel, or on your TV. When do we need to use it? First of all, when we have very viscous systems, which cannot be made into the thin films that are used for TEM. Then in some cases, and this is very relevant to our case, when we want to see fine details in large objects. If we want to take a whole cell, the cell is several micrometers across, like blood cells, and this will not fit into the very thin specimen. So we have to use a different type of instruments. And in this case, as I said, we use the scanning electron microscope, and nowadays we can use a high resolution Cryo-SEM, which has very sophisticated detectors and a field emission gun for emitting the electrons, with the advantage that we can use very low acceleration voltages of about 1 kV, which is very useful for our microscopy. This is how the instrument looks. It looks much smaller, again, for reasons that I'm not going to go into. And let me just show you a couple of examples of what we can do. First of all, how do we prepare the specimen? Typically, we use what we call the sandwich technique. We have two gold planchettes or two gold cups, and we form a sandwich. Here you have a schematic of it, where a small volume of the liquid, a very viscous liquid sometimes, is held. We then plunge this sandwich into the cryogen. And after that, and you can see it here in the schematic, we open the sandwich and we form the two fracture surfaces. And these are the surfaces that we are going to examine in the microscope. Again, without going into details, more than 10 years ago, we were able to develop an analogous controlled environment vitrification system for Cryo-SEM, which means that we can prepare the specimens under controlled conditions of temperature and humidity in order to avoid any loss of volatiles from the specimen. And just two examples, and this goes back to a paper by Naama Koifman, who is in the audience now. And here we have a very nice monocyte, and you can see here the entire monocyte here. And this is the nucleus, and then here you can see the entire cell. In this case, the cell itself was fractured, but the fracture surface went over the nucleus, so the nucleus itself was not fractured. In some areas here, you can even see the nuclear membrane with nuclear pores. In this case, the fracture went through the nucleus, and you can see very well the fractured nucleus and the fractured cell. This is probably the most important picture because I think this relates to slide 24 of Kelly's talk, and what you can see here, and this is what Naama did. She was able to actually capture the process of the budding of the extracellular vesicles. So you see here you have some of the beginning of the buds. Here you have some completed buds, and some, in fact, EVs that are completely free. And oops, sorry. Okay, so again, just a last remark, here you can see very nicely the nucleus with the nuclear pore seen very, very nicely. I forgot to just mention that all the nice data of the bacteria was done by Irina Davidovich, who is also in our audience. So this is just a very short sampler of what we can do by electron microscopy, and I think I will finish at this point. Thank you very much.
Hey, thank you so much, Kelly and Professor Talmon, for that fascinating account of the published paper and the techniques. And I think it's really incredible. I mean, this is a piece of the puzzle that has maybe been missing from the field. Not that it wasn't there, but perhaps that it just went unnoticed or unregarded for quite a while. And it's definitely going to open up a big can of worms for everyone, I feel. So we do have a question in the chat box, but I would like to ask one question of my own before we get started. So Kelly, this might be something that you might be better off answering, as I think you touched on it in your presentation, about the membrane damage that may contribute to formation of the multi-layered EVs. And I just wondered, is this potentially a homeostatic mechanism? And what I mean by that is lipid swapping or integration with the extracellular environment in response to membrane damage that could cause these multi-layered EVs. Does this kind of imply that perhaps EVs have a potential mechanism of repairing themselves to some degree?
Yeah, that's a great question. That's actually something very early on Joy and I discussed when we had these images and these perturbed THP1s with increased membrane damage, but then generating these multi-layered EVs, whether it was a repairing process, and we don't know, but it feasibly could be. It would take some clever research, I think, to figure out if it is, but I think it's a very interesting line of inquiry to pursue further.
Yeah, definitely. I think it's fascinating. So I'll get on to the next question so as to not take over. So we have a first question from Duran Balan. Would you like to unmute yourself and ask the question?
Hi, can you hear me?
Yes, yes.
So my question is that, is the existence of multi-layer vesicles a hindrance to cargo removal or loading them?
Yeah, so that's a really interesting question, and it kind of ties into what the function of these structures actually is. If it is in response to damage and it's the vesicle repairing itself, it could very well be a hindrance to unload whatever is in the vesicle, whereas if they're intentionally being created through some biogenesis mechanism to be multi-layered, it may be intentional and less of a hindrance and more of a benefit. But there's so little known about the biogenesis pathways of these vesicles that it's really difficult to say for certain whether this is more of a positive or a negative physiologically. And I think that really depends on what's happening and what the context is, whether it is a hindrance or a benefit.
Okay, thank you.
Great. And I'll open this question up to all of the authors. This is just another question following on from the last one. So have you discussed techniques to possibly study these multi-layered EVs? Like, how would you even begin to isolate the multi-layered EVs away from the single membrane? Staining techniques, kind of mixing one stain in with another stain and putting together and seeing if you can co-localize like an antibody or a fluorescent probe under confocal. Like, what kind of things have you discussed with regard to digging into actually studying these things?
Okay, well, this is not directly an answer, but you have actually mentioned the possibility of labeling the system, and this is a very important issue. In fact, one of the pioneers of the labeling by immunogold labeling in the liquid phase, not in the dry phase, the liquid phase, is here with us in the audience, Alain Brisson, and from Bordeaux, and he really pioneered this. And we have tried to take this one step further. We, in this case, is Naama Koifman, who is also now in the audience. And we are in the final steps of preparing a manuscript about that. Now, this should answer many, of course, if Naama ever sends me that final version. But this is really an important thing because, first of all, if you do it properly, and it's really not a trivial task, you can try to find what type of lipids or other molecules you have in the membrane of your EVs. And by doing this, you can go back and look at where was the origin of that. Now, this may not allow you to filter out only the single membrane particles from the multi-layered vesicles or multi-layered EVs, but it will definitely give you more information from where those came. And this can also shed light whether you have actually done something to them during the preparation.
That's wonderful. That's such a great explanation. I guess I'm very excited to see the manuscript as well when it comes out. But Alan, I think you actually put something in the chat box. Did you want to have a little discussion around your comments on the multi-layered EVs?
Yes, of course. Yes. Well, hi all. Hi Ishi.
Hello.
It's a bit early here, so my voice is a bit bad today. Yes, a comment about our experience with Cryo-EM, mostly based on, let's say, on fresh plasma to make things simple. We see, of course, multi-layered EVs, and I would say for most of them, they look like pure lipid vesicles, and Ishi has shown some classical, say, liposome, multilamellar liposome, and they look like that. What I mean by that is that they look transparent and very different from most EVs that contain something inside. They look more or less gray, more or less dark, depending on the amount of material they contain. While multi-layered EVs, for most of them, I say, they look transparent. Basically, there are no surface, no membrane proteins protruding at their surface. Well, with the resolution we have with a microscope, but say an expert eye is able to see those protrusions. And about immunogold labeling, with all the gold labels that we have used now, which are of course specific to blood cells, strictly no label and no phosphatidylserine exposure, for example, also. But this is one class of multi-layered EVs. We consider a totally different class are those, for example, with activated platelets. Activated platelets may be as large as a micrometer and contain different, say, mitochondria, granules, are labeled with CD41 or annexin 5, for example. They look totally different. So I have a question here for you. What about, is it your experience that those multi-layered EVs you are working with look like pure lipid vesicles?
Not all, if I may, not all of them. I think it's a very good point, Alain, and I think many of them look like pure, but some of them you do, in some cases, you do see granulated liposomes enclosed within a pure liposome on the outside. Now that you have made this point, I think we should go back and look more carefully on our results. But you have to remember that we looked at a very wide range of sources of EVs, and different sources may actually give you different types of EVs. But this is a point that should send us back and check our data. Thank you for making it.
Good. Go back to work. Good.
Thank you.
There's always more work to do. And I would say, absolutely. I mean, we see that time and time again in results, EVs from different cell lines, from different biofluids. They all seem to have very unique characteristics. So one could assume that it might exactly be the same for the multi-layered EVs of different types, different cell types, different fluid types. So very interesting points. Does anyone else have a question from the audience that they'd like to put forward at this point?
Sorry, I'm about to teach, so it's a bit loud. Great presentations. I know that, for example, Nam and I have discussed like the gold labeling, immunogold labeling could be dependent on the curvature. So if there's larger EVs, then the curvature would be less and maybe it would be more successful. So I'd love to hear the experts here in the audience talk about that topic. Thank you.
If I may say, answer the question about the influence of curvature on gold labeling, it says this is not what we observe. And say from small EVs, say 70, 80 nanometer to micrometer large EVs, we see about phosphatidylserine exposure and Lexin-5 labeling. There is no, I would say, no, it's not quantitative totally, but there is no difference. The same with CD41, the same with CD63. So I doubt that curvature has an influence on immunogold labeling, at least with those we have analyzed.
Thank you.
So I think in the chat box, was it Uta? Did you have a comment?
Yes, sorry,
Perfect.
it's already
Thank you.
late here.
Thank
you for the great discussion. I just want to make a comment that I don't know if it's an observation. After this, I will look more into our pictures, but I feel like we see them in clusters, you know, if that's another phenomenon criterion. And we have seen them or generated them from preps, which have been frozen before, and in different types of biofluids, urine, cell culture, so I can all confirm that. Yeah, thank you. And I'm curious to know what function they have, if that exists. I like your discussion.
Yes, perfect. That is going to take some very special handiwork to try and figure out what these specific multi-layered EVs are doing differently to single-layered. Yeah, again, I'm intrigued at these methods of differentiating them. I just have to wonder, given the massive amount of literature that has been produced in the last decade or so in the EV field, and knowing that Cryo-TEM has not been readily available for a lot of researchers, how many studies have been performed that have included, you know, the whole EV populations and unknown percentages of those multi-layered EVs, how much of our results are attributed to multi-layered EVs already that we just don't know. It could be significant given your findings and the literature review that you've done for this letter to the editor. And pathogenesis or normal cells, are they producing different amounts? I think, Kelly, you showed that it was true in both cases. And who's to say that a healthy cell isn't producing a large amount, or unhealthy cell producing a large amount? Without these techniques to properly separate them, it's going to be really challenging. So I do look forward to more advances coming soon. So if anyone else who would like to put a message in the chat box, Mohammed, I think you've put a comment in there. Would you like to unmute yourself? Sorry,
I'll be
Nama?
happy to say something. Yes.
Oh, please do. Yes.
So thank you for the lovely presentation. Thank you, Ishi, and thank you, Kelly. I enjoyed it a lot. I would like to suggest something here, as we are quite a big audience of people who are interested in EVs, and a fair amount of people also doing cryo on these. One of the things I'm experiencing now, I'll just present myself. I was a PhD student of Ishi, and I've graduated, and I was the center manager at Technion, but then I relocated to UQ, and currently I'm working here at the Center of Microscopy and Microanalysis. And I do quite a fair bit of fee-for-service work on EV samples. And it's very interesting because I get exposed as a person who provides these services to different samples with different characteristics. And one of the things I think that would be important in order to advance the field, and specifically because Cryo-TEM is such a powerful tool to investigate these type of samples, is maybe as a community of researchers to try and establish a kind of a database. Similar to PDB or other, like maybe the parallel ones would be the ones that protein researchers have, where people can share maybe after publication the raw data. And then because the study of EVs, it will rely, I think, in the future, and there's enormous amount of data being collected. The only way to actually study them in large amount would be to be exposed to and to compare different data from different sources. And I think if we as a community can push this idea forward, I think that would be very beneficial to understanding differences and similarities from different resources. So I'm just putting this forward here, and anyone is interested, please let me know.
So. Nama, I think there's been affirmative from Joy and Ken in the chat box already. So
yeah. I've discussed that. I've discussed that with Joy and with Ishi. But I think using this forum, I would be happy to have more people on board and maybe try and do something that would be good.
Excellent idea. Thank you, Nama.
Thank you. Thank you so much for those comments. It's really helpful to have someone who is really hands-on looking at samples all the time, as you say, fee for service, so you're not seeing the same thing all the time, to have those eyes on and give that experience and to know what you're seeing. That's fantastic.
No, and I think, yes, and I think if, that's the nice thing. So, for example, Professor Brisson is looking at samples that are mostly from, let's say, from blood plasma, and other people are looking from cell culture, and other from kidneys, and other from urine and semen. And these are all, yes, there are similarities, but in order to understand what these are, you really need to go over a large amount of data. And I think with the progress also with image processing, maybe possibly in big data and AI and all these advancements, maybe if we can use all these tools also to help us understand this system.
Thank you. Yeah, fantastic. Great idea. Great comments. So we are now at the end, I think, of the presentation and the discussion. Again, thank you to everyone in the audience, all the co-authors and authors who joined us today. It's just been the most interesting topic to discuss and so much to learn. So thank you all, and thank you, Joy, for organizing between all the different locations. So we'll leave it there. Thank you all very much for participating today, and I hope to see you at ISEV.
And thank you, Natalie, for moderating it so beautifully. Thank you.
Yes, thank you.